Hello everyone and welcome to the 8th of this year's webinars, today on the topic of the role of bioinformatics in proteomics. We're joined by two speakers, Dr. Juan Antonio Vizcaino and Dr. Oliver Crook, alongside our guest chair for today, Professor Andrew Dowsey. Andrew will chair the question and answer session after both talks in a roundtable discussion with both of our speakers. So I now just have a few of the usual housekeeping points to go over before handing over to our guest chair to introduce the first speaker. As always, we're using our Slack channel for questions and discussion, so please join us there to ask questions and use the thumbs up to let us know which questions you'd also like to hear answered. Please direct your questions to each of the speaker by naming them, as we'll be having the joint Q&A and roundtable after both talks are finished. For those of you needing an attendance certificate for this webinar, details will be available on how to get this on the slide. And we'd like to say a big thank you to the European Proteomics Association, the British Society for Proteome Research, the Young Proteomics Investigators Club and the London Proteomics Discussion Group Committee for their help and support in setting up this webinar. Thanks also to the London Biological Mass Spectrometry Discussion Group, the London Metabolomics Network and the News in Proteomics Research blog for promoting this event. We're also grateful for Imperial College London for providing us with the webinar support. Thanks also to our YouTube channel subscribers. Both talks today will be available to watch again online. So a huge thank you to our speakers and guest chair for their time today. And now Andrew will introduce our speakers for today. But first, just a quick introduction. So Professor Dowsey is a chair in population health data science and chair in One Health bioinformatics and biostatistics at Bristol Veterinary School. His research group focuses on the facilitation and acceleration of health sciences research through novel artificial intelligence, physical data science, as well as data collection and management platforms. He is co-director for Enterprise and Innovation in the Faculty of Health Sciences, is an associate director of the HDR UK Southwest Better Care Partnership Fellow. To introduce the first of today's speakers.
Hi everyone. Thanks very much, Joan, and thanks very much for inviting me, and thanks everyone for turning up today to hear a couple of fantastic talks. I'm first going to introduce Oliver Crook. He's a postdoctoral researcher in the Department of Statistics at Oxford, and he's a Todd Bird Junior Research Fellow in Biochemistry there. So he was previously a Wellcome Trust PhD student in Cambridge, and many of you on the call will know that the people he was working with, Catherine Lilly and Laurel Gatto. So it's my pleasure to introduce Oliver. He's going to talk about quantifying uncertainty in mass spectrometry based proteomics with Bayesian methods, which is also close to my heart. Take it away, Oliver.
Hopefully I'm unmuted and you can see my presentation.
Yes.
Great, so I'll just get started. So one of the kind of fundamental questions we ask ourselves in proteomics is my protein differentially abundant? And this is the kind of a key experiment that many of us will have performed or want to perform, or will have got someone else to do for us. But the answer will depend on many things, such as, what type of mass spectrometry I did, so whether it was a label-free experiment or a TMT, or whether I used a Waters instrument or a thermo instrument. Obviously, the context of a protein being differentially abundant will be in the context of the biological system. So was it yeast? Was it human cells? What perturbation did I use? But then there are kind of more technical questions like, did I identify that protein correctly? Was it the one that I thought it was? Did I pick a number of thresholds in my analysis to kind of filter out certain results? What were the settings that I used on the mass spectrometer? Did I inflate missing values for some reason? And you might want to include all of this information in a particular analysis, and that leads us to how do we model all of these things, and then ask the question, okay, given all of this information, how would we calculate a probability that my protein of interest was differentially abundant in that experiment? And all of these experiments that we do in mass spectrometry, you will do your own version. I imagine most people focus on one particular technique or a suite of methods to answer a particular question. But we have to think about what choices we make in those experiments in the experimental design, and how uncertainties arise in each of these. So I'm going to mostly talk about things from shotgun proteomics, spatial proteomics, thermal profiling, so TMT-based things, but also hydrogen deuterium exchange, so and DIA-based analysis. But please just think about how you do your experiments and kind of map on the ideas as I go through. So often we'll use mass spectrometry to acquire some data, and then we'll probably go through what many will find as seemingly magical filtering steps or processing steps, and everyone will do it slightly differently, but there are many things that one might do. And after going through these steps, we've got this nice shiny data set at the end, and most of you will want to perform some kind of statistics or data mining or machine learning on that data. Why do we go through all these kind of seemingly unrelated or perhaps confusing or kind of highly variable steps? Well, there is kind of reason to it. We've probably got a huge data set, and so summarizing it saves time. And ultimately, we hope that it leads to a kind of smaller, potentially more confident set of proteins and peptides. There's no point making conclusions about very noisy data. And we're also probably not going to lose power in something like multiple hypothesis testing because of low quality data. We filtered it out long before it got anywhere near any statistics. And the thing that I worry about is that any one of us may make different choices, and in many cases I come across these questions. So how reproducible are our experiments, and how much variation is as a result of these kind of processing steps that we've put forward? Would we find support for a different biological result as a result? And are we often kind of missing important insights because of these choices? If we throw data away, what does that mean for our downstream results? And perhaps we might even do the experiment differently if we were able to characterize our uncertainties at each step and then get the final output, and that may inform kind of ultimately the experimental design much better. So we need some tools to do this analysis. Unfortunately, I'm only going to briefly talk about Bayesian statistics, but what it starts with is a model for our data, often encapsulated in a likelihood function, and this is coupled with a prior distribution of the parameters of that function. And together you can think of the prior and likelihood together as describing some kind of generative model, so a way of generating data. And so once we've observed data, we can update our prior distribution into a posterior distribution. And what that says is, gives me a probability distribution over the parameters or quantities of interest. And that update is performed using something called Bayes' theorem. So it's just an equation that tells us how to perform that kind of rational update. Maybe some key things to note about Bayesian statistics that might be different from how other frameworks you might have worked with is that kind of simulating data or predicting new data involves integration and averaging rather than taking one value forward. We do this analysis over all possible plausible values of the parameters, kind of weighted by their probability. And that's true if we're simulating data, but also if we're predicting data, and we can take the posterior distribution of our parameters and then simulate new predictions from that. So the first example I'm going to talk about is very close to what I did in my PhD, is the example of spatial proteomics. So in these experiments, we start off with a population of cells, and we're going to gently lyse them in such a way that the organelles remain intact. And then we're going to separate them biochemically in some way, so either using density-based centrifugation or differential centrifugation, or some other kind of detergent-based biochemical fractionation. And hopefully these organelles will separate along these biochemical fractions, and then we can collect samples from this in this case, we use TMT to multiplex these, and then you can perform downstream mass spectrometry. And the key insight people have made is that if proteins localize to a particular organelle, they share a unique signature along this density gradient. And so, for example, all proteins belonging to the mitochondria will have this kind of pink signature that you have in one of these panels. And so what I can then do is employ machine learning or statistics to predict proteins with an unknown location to one of the organelles in the cell. These experiments have come a little bit further in recent years, so we can do this not just to localize proteins, but also look at differential localization. So how do proteins move around the cell as a result of a perturbation? So we essentially do that protocol that I just mentioned, but in two parallel frameworks. So you would have a control and a treatment, or condition 1 and condition 2. You perform those experiments and then do downstream mass spectrometry. And so what will happen is if I've got a protein, for example, that relocalizes from the plasma membrane to the lysosome, it will change its signature. And hopefully that's the idea is fairly obvious from the first figure. A protein that doesn't move will just have some kind of stochastic variation and look slightly different, but hopefully not too much. So how do we analyze these data, or what do we want to get at? So ultimately, we want this quantity, a differential localization probability. And for each protein, I'm going to ask, I want a statistic that tells me, is it differentially localized or not? And we're going to define this probabilistically as the localization in dataset 1 is different from the localization in dataset 2. And I want the probability of that quantity. So things with probability 1 are going to be differentially localized and 0 not. They will remain in the same place. And what are the kind of parameters that we're going to think about modeling here? Well, we've got differences between replicates, so we should allow some variation there. And we've got some uncertainty in those marker distributions. So those might change, those signatures might change between experiments. And so we can start putting together a model for these data. So what are we going to start with? Well, we're going to think about each replicate separately and model those signatures along those fractions. And then generate a model for each replicate in each condition separately. Then you can start thinking, okay, I can integrate the models for each condition and get an allocation for where that protein or which subcellular niche that protein was in that condition. And then using a joint prior model over these two conditions, I can calculate a probability that a protein was differentially localized or not. So this protein seems to localize to subcellular niche 3 in one condition and subcellular niche 2 in the other condition, and hopefully this model will be able to capture that. And so what this allows you to do is rank your proteins according to that probability. So probability 1 or close to 1 will signify that it's differentially localized, whereas proteins with lower probability are likely spatially stable, and it was just kind of noise generating any differences. But because we did a Bayesian analysis, we could also get uncertainty in that measurement. Okay, but why do we do all of these things? Well, modeling that uncertainty and capturing that phenomena of interest actually does a much better job of identifying proteins that we believe are moving, or what the ones we're interested in, and compared to kind of ad hoc scores in the literature, which are kind of not generated from a model and not thought about what are the variations in these data sets. So as you can see from this violin plot, the signal is much clearer. So what about another example? So that is of thermal proteome profiling. So in this case, we take populations of cells again, and we might treat some of those cells, for example, with a ligand, but it could be any kind of perturbation. And we're going to take that population and use sequentially heating temperatures for that sample, and then extract those samples at each temperature and perform, again, multiplex and mass spectrometry. The hope is that proteins will denature during this process, and you're just measuring the fraction that was non-denatured during that. And so if a ligand bound to a protein and thermally stabilized it, then it would have a different kind of mass spec signature along these, according to these, as a function of this temperature. And so this will allow us to identify proteins to which the ligand bound, but potentially off targets of that ligand, or even downstream perturbations of that system. And kind of biophysics tells us that the data analysis should revolve around a sigmoid model. So we can just fit a 3-parameter sigmoid model to these data and then perform standard statistical hypothesis testing. But I know this is a choice, a modeling choice that we choose. And how frequently does that modeling choice play out well in practice? So I had a quick look at some thermal proteome profiling data, and I found that, you know, actually quite a lot of the time that sigmoid model is really a great model for these data, and the everything we expect to be true is true. But in some cases that's not the case, and here are some examples of just some random proteins where you can see nice sigmoid behavior, but also things that clearly do not look like that beautiful S shape. They're clearly deviating in some way. And so how can we model these deviations? Well, we can use a technique called semi-parametric modeling. So we can take our TPP data, so we have some response, and then use that sigmoid model we had before, but then also add in a kind of non-parametric term in the model to add in some flexibility. And what this is going to do is you can think about scooping up any residual unmodeled noise that was left after taking away any sigmoid part of it. And so, for example, you see this curve in purple couldn't possibly be modeled by a sigmoid because it has 2 inflection points. But our kind of semi-parametric modeling framework can easily handle such a case. So because we're working in a Bayesian framework, we can literally ask if the semi-parametric modeling framework actually models better any of the data than the sigmoid model. And from there we can say, okay, are these proteins that deviate from that sigmoid model special in any way? And what we found was strong enrichment for proteins of phosphoproteins, proteins involved in RNA binding, and proteins enriched for that had localizations according to the mitochondria and nucleolus. And this is across the whole spectrum of possible functional annotations these proteins can have. So it's not really clear what's happening. All we can say is that, well, they're functionally related in some way, and you'd have to perform kind of many more experiments to understand why this is the case. Why do they have these deviations? So the final example I want to mention is hydrogen deuterium exchange. And so in these experiments, it's usually performed only with a single protein or a small protein complex. Proteins are incubated in heavy water, and what happens is that the hydrogens are able to exchange with deuterium. This is done over a time course, so proteins, hydrogens that are easy to exchange with deuterium on the kind of surface of the protein will do so quickly, but it will take longer for buried hydrogens to exchange. So this gives you an idea or some proxy for protein structure. So this reaction will be stopped. We use a peptic digest, and again these peptides can be measured using mass spectrometry. And the key thing that we're looking for is that as peptides incorporate deuterium, they're going to have a positive mass shift because deuterium is heavier than hydrogen, and they're also going to undergo kind of isotopic expansion. And then we can measure deuterium incorporation over different times, hopefully in different states or perturbations to our protein, and that can help us understand dynamics of our protein. So again, we can apply a lot of the tools we have in our previous analysis, so spatial proteomics and thermal profiling, and just posit a model for these data. So here we have time along the x-axis and deuterium incorporation along the y-axis, and we can say, well, let's model this data. In this case, it's a kind of logistic type model, and we see here the state is whether an antibody is binding to this protein or not. And as a result of this antibody binding, we have increased deuterium incorporation. So that suggests this protein has a slightly different conformation bound to this protein. But we could have done this pretty much with any tools. Are there questions that more complex questions we can ask of these data? And one thing we really struggle with in HDX is the idea of significance. How much deuterium incorporation is actually interesting? So what we can do is calculate the probability that the change in deuterium in two states is greater than some threshold or some value d, and then ask only for peptides where that probability is itself greater than 0.99 or 0.95 or 0.9, and then calculate the minimum deuterium incorporation we're allowed to ask for to say something interesting. So for example, your eye is probably drawn to this peak kind of midway on the right hand side, and that's kind of maybe that we should be looking here, and the rest of it is mostly noise. So this was for different probability thresholds. We can also ask, how do these dynamics change with time? So instead of plotting different probability thresholds, we can plot different exposure times, and again be drawn to different styles of kinetics and examine exactly what is going on with this protein at particular times. And these are computations that you wouldn't be able to obtain within a frequentist framework or a different modeling framework very easily. Maybe some food for thought is, in a lot of these experiments, we're summarizing our data down to something. So in HDX, we often take a spectra, and the data we're using is this kind of centroid in orange, but this spectra on the left-hand side is very noisy. There are a lot of clearly irrelevant peaks going on. And on the right-hand side, we have a much cleaner spectrum, and clearly we have a lot more confidence in that centroid or orange value. But this is completely ignored currently in the analysis we're doing. And should we be doing something different? So I'm going to wrap up and say, well, have I really answered the question that I set out to? I gave you all these parameters and say we should use all of these things to develop a model for our data and calculate a probability. Well, no, I haven't used all of those parameters and data that I could. And I say, well, I'm still working on it and I'm only one person. But I wanted to say that there are essentially strong hints that incorporating even a small amount of this uncertainty is very useful and clearly very powerful approach. So maybe we should be doing more of these things. And finally, we can obtain information that would be very hard to any other way. So hopefully get more insights from our data. Thank you.
Great. Thanks very much, Oliver, for a fascinating talk. And I've got some questions for you later, but I'm sure others have too. So please submit your questions and we'll ask them and discuss them after Jaun's talk. So I'd like to next introduce Jaun Antonio Vizcaino. Jaun is team leader for proteomics at the EBI. His team are responsible for the PRIDE database, and he leads the Proteome Exchange Consortium. I myself know Jaun a long time from both our involvement in the Proteomics Standards Initiative. So Jaun, please take it away.
Hello, Andy. Can you hear me?
I can.
Okay, perfect. So what I cannot see is still my slide because the request for control was not accepted.
Oh, there we go.
So yeah, I will do
Sorry,
it.
Juan, can you ask again? There you go.
Yeah. Yep, that's fine. I can see it now. Okay, so again, thanks to Andy for his kind introduction, and also thanks a lot to Harry and all the organizers for having invited me to participate today in this webinar. So my name is Juan Antonio Vizcaino, and I'm based in the European Bioinformatics Institute, very close to Cambridge. The title of my talk is Bringing Proteomics Data Closer to Biologists, Disseminating and Integrating Public Proteomics Data into Added Value in Bioinformatics Resources. So this is the overview of my talk. I will first give a short introduction about PRIDE and proteome exchange, and then I will talk about the main topic of this talk, dissemination of proteomics data to added value resources. So the PRIDE database is part of the portfolio of bioinformatics resources that is provided by the EBI, so the European Bioinformatics Institute. I'm sure many of you have used resources like or PDB or Reactome, and actually the EBI provides probably the largest collection worldwide of bioinformatics data resources. Of that is PRIDE, who was started in 2004 by Brickner and Martens, and PRIDE stores mass spectrometry-based proteomics data sets coming from the community, storing different peptides data, and basically all proteomics approaches are supported. And just to give you an idea of the amount of data that PRIDE stores at the moment, as of October 2021, it stores more than 24,000 data sets. It is important also to highlight here that as a community, we have been able to coordinate efforts for submission and dissemination practices in an international manner worldwide. I think we should be proud as a community to have achieved that. And we started the Proteome Exchange Consortium in 2011 to standardize these practices. At the moment, there are 6 members in Proteome Exchange. PRIDE is the European representative, and then we have 3 colleagues in the US, Peptide Atlas, Massive, and Panorama Public. And then we have 2 colleagues, 2 member colleagues in Asia, IPROC. And as you probably are aware now, data sharing has really generalized in the proteomics field. There has been a push by journals, by funding agencies, but also because open science practices are a good scientific practice. So just to give you an idea about the growth of data in PRIDE over the years, just a chart that shows the growth over the last 10 years is not cumulative. It's kind of month per month. So you can see the really dramatic in data that PRIDE receives every single month. So the record of kind of submitted data sets so far was on March this year, when we received almost 600 data sets. But just to give you an idea, in the last year, PRIDE has received on average 500 data sets per month. That's really a lot. And it's also good to highlight, I mean, PRIDE is a resource base in the UK and also in Europe that is really the world leading resource in this context, storing more than 83% of all proteome exchange data sets. Something also that I want to highlight is that PRIDE is the third resource in size in EBI after the ENA and EGA, which are resources devoted to DNA and RNA sequences data. And you can see here the huge, again, increase in data sizes. And just to give you an idea, just to finish about this part, 44% of the data sets are from human origin. And then, of course, the usual suspects, the main model organisms, mouse, Saccharomyces, Arabidopsis, rat, etc. But there are actually data that are coming from more than 3,000 taxonomies. So even if you are working with, maybe an organism that not many people are working, there is a good chance that there is some proteomics data in PRIDE from that organism. Okay, and I just wanted to finish this kind of introduction to PRIDE, highlighting, I know that this webinar was devoted for quite a few weeks, months to COVID-19 related topics. So the EBI set up the so-called COVID-19 data portal. This is the URL. that combines data coming from related to COVID, of course, coming from many different origins. And among those origins, you can find proteomics data in the expression part of the portal. So just in case you didn't know this, it can be also useful for your research. Okay, so let's go for the main part of my presentation, dissemination of proteomics data to added value resources. So, of course, what is the objective of having all this data available for everybody? So the objective, apart from being used to assess the results that are, the main objective here is that the data is reused and that new knowledge is generated. And this is happening more and more. There are more and more publications all the time about this. It is used in benchmarking studies, meta-analysis studies, proteomics, PTNs, data integration, many different efforts and approaches. Many of them are novel, and this is very nice to see. You can also see here the growth in volumes of data downloaded from Bright since 2013 to 2020, and you can see there is a huge increase in recent years. I think that one of the things that we should be aiming to do with this data, and this is kind of the main topic of my the main take-home message of this presentation, is that the data that is made available by the community into proteomics resources, in this case Bright, it is good to reuse it as much as possible, so that it can be we have a better representation of data in bioinformatics resources that are used by many biologists, and many of them are not really familiar with proteomics. So these are some examples like UniProt, Ensemble, GPNDB, ProteomicsDB, but there are other ones that are kind of outside of the field, devoted to proteomics, and I think this is really important for us to get more prominence as a. So in this context, the team, we have been working in these efforts in recent years, and because it's of course easier for us to do it this way, and also because in the ABI, as I mentioned before, there are resources that are very widely used by the life science community. What we have started to do in recent years is to integrate proteomics data with other omics data types that are in popular bioinformatics resources in the ABI. So we have done that for protein sequences and PTN, UniProt, as I will show, protein expression information. The resource that we have been collaborating with is Spatial Atlas, and then also proteomics data, Ensemble in this case. And very recently, and I will not be talking about this in this talk, we have started to integrate metaproteomics and metagenomics information in the Magnify resource, which is the ABI resource devoted to metagenomics information. So the main approach that we have followed so far is based on data and analysis, and this is what is being made or is being done mainly in the community by our colleagues of Peptide Atlas and GPNDB for many years in order to make data more comparable. And we have been doing that in recent times, as I mentioned before, with Expression Atlas, with the idea of integrate gene and protein expression information. It is important to highlight that Expression Atlas is an EBI resource that has been accumulating gene expression data for many years. First tissue, or what is called Atlas, and lately more focused on single cell expression information. So now quantitative proteomics datasets are systematically reanalyzed, integrated in Spatial Atlas, so they can be accessed by everyone. So we have done so far this in a project basis. I'm not going to explain all the details, but I want you to get the idea. We have been doing this for data-dependent acquisition data sets. We have done 3 main studies so far: cell lines and tumor tissue data, human baseline tissues, and mouse and rat baseline tissues as well. The selection criteria are data sets coming from thermoscientific instruments, just to make data more comparable, level-free experiments, no enriched samples. We are not interested on PTNs at this point, and with enough metadata. And this is really a key point, that data needs to have enough metadata in order to be, a consistent reuse of it. So I'm going to talk a little bit about the work that has been done by Anand Prakash from my group that has been working on generating expression data coming from human baseline. So the main take-home message here is kind of the protocol that needs to be followed. First, the datasets need to be manually selected. In this case, we use datasets from Pride, but also from other sources. In this case, we also got brain tissue data coming from the EMBL knowledge portal database. It's an American database devoted to Alzheimer's disease. And then what it needs to happen is that once selected datasets, they need to be curated, adding the necessary metadata annotation. And that metadata annotation, it can be quite time consuming to get at this point. It's something that we would like to change iteratively in the future. And then that metadata needs to be put in the right formats, in this case, SDRF and IDF. So we started this project in September 2020. At that point, we found almost 3,000 datasets that were suitable for that based on criteria that I told you before. And at the end, we performed this analysis that comprised 32 organs, 68 tissues, almost 5,000 MS runs, 1,500 samples, and a little bit more than 1,500 individuals. So, basically, after the selection, there's the part of the analysis. For DDA datasets, we use MaxQuant in a HPC Linux environment. We use the UniProt human reference protein for that. Then after that, we do some downstream post-processing, like normalization of the protein abundances. We also map the proteins to gene identifiers, and we also do a further bin transformation of protein abundances to make data, again, more comparable. And then the last part is the kind of production part that we in the ABI can take care of. That is basically the integration of the data from Pride into Expression Atlas. And we also make the data for everyone and to produce a quality assessment, quality control summary of the datasets to be completely transparent to everybody. Okay, so we have done that for data-dependent acquisition, and we have also started to do that for data-independent acquisition datasets. This is a work that has been led by Matthias Wolzer in my team. So in this case, this is technically much more challenging than DDA datasets for several reasons. So so far we have done a kind of pilot study using lines and tumor data and also serum data. And the selection criteria in this case, we have used data coming from SciEx instruments only, so SWAT-MS so far. Again, metadata so that we can make sense of the results that we want to communicate, and that all the data types needed for the processing are available in the public domain. So again, this is the pipeline that we have used in this case. Again, the first part is the manual selection of the datasets and the curation. This is not avoidable, and it is really a big chunk of the work. The last part is the kind of loading of the information into Expression Atlas. The part here that's a little bit different is the actual pipeline, which was developed in Nextflow using OpenSWAT and related software like PyProfit and Trick. And then there is a second part that is related to further normalization of the results. So I don't have time to go into the details of the findings here, but I want to tell that this information is accessible in Expression Atlas, integrated in the web interface. You can see gene expression and protein expression data. This is just one example of a query using this UniProt identifier. And if you zoom in a little bit more, you can see that, again, it is possible to see coming from proteomics here, together with data coming from transcriptomics. It is possible also to access the individual data sets individually as well. I want to highlight that we have done this in a kind of conservative manner. The objective here is not to show kind of every last evidence of expression. We just want to really report the results that are reliable. So this is what we are doing. Okay, so if you want to know more about this, there is one paper that was earlier this year about cell lines and human tissue, and then there are 2 preprints in bioRxiv about the analysis of DDA and DIA datasets. So, in addition to Spatial Atlas, another resource we are working together is UniProt. In this case, the objective is to improve post-translational modification data that is available in UniProt, and to improve the traceability between the PTNs annotated there and the primary data, basically the MS data stored in Pride. We have, of course, done this so far only for phosphorylation, the most popular PTM. And in this case, we are working together in a project with our colleagues in ISB in Seattle, a Deutsch long-time collaborator, also Andy Jones from the University of Liverpool, and my colleague Maria Martin in EBI. What we are doing there is we are selecting manually data sets that in this case are enriched for PTNs, for phosphorylation, and then we are running through analysis pipelines. Mainly at the end, we are using a pipeline that is based on TPP software, and then what we are doing is generating pipelines to disseminate this information from Peptide Atlas and Pride into UniProt, and to make, again, this information as transparent for the user. So I cannot show you anything working yet. You have to believe me that we are working on this, and hopefully I think at the beginning of the next year, the first kind of linked data sets will be available for everybody to see. Okay, so a third resource we are working together is Ensemble. The objectives here is to integrate efficiently high throughput proteomics, and in this case, peptide sequences can be integrated in Ensemble, but also in other genome browsers using the so-called TrackHub mechanism. That is a technical infrastructure that can be used for this purpose, to basically access to external data in genome browsers. And the reanalysis in this case is, again, project based, and is done in collaboration with Jyoti Chudari's group in the Institute of Cancer Research. So this is kind of one project. This slide is courtesy of James White and CR there. And what we are doing is using cell lines, on one hand, public data from the Cancer Cell Line Encyclopedia, from the other hand, in-house data from colorectal cancer cell lines. We are using a pipeline that, again, has been built in Nextflow using OpenMS, and we are using databases to detect as many variants as possible. And again, all this information will be made available via Pride and link it to Ensemble as well. So again, I cannot show you a final result of this because this is work in progress. But again, we hope that this information will be again soon available for everybody to see and connect. The final result of this, 1 second. Yeah, sorry. Yes, sorry. I will go back again. Is that again, peptides can be visualized in Ensemble web interface. This is just one example. Or we can see there the peptide in the context of other genomics features. And just to finish with this part of analysis, we are not the only ones who are doing this. There are new resources that are being kind of set up based on the reanalysis of public, and these are just some examples. So that have kind of been started in the last couple of years. So Proteomics or SCOP3P for post-translational modifications, or MatrixZombie, OpenProt, which is a proteogenomics resource, or MagenDB. So these are all the resources. All of them are published, and these are the corresponding. So you have some interest in accessing them. This is great because Proteomics public data is more and more reused. And the last part of my presentation, I'm going to talk about the other part of the work that we are doing to add better resources. In this case, based on an automatic pipeline using quality filters submitted data. So all the work that is based on data analysis is really very resource intensive. A lot of resources needed. people to create the data, to analyze the data, to is really a lot of work there, and to have an automatic pipeline that moves the high quality submitted data from Bright into and make it linkable to other resources. This is work led by Jacet Perriverol from my team. Resource, well, by Proteomics, but has been changed completely recently and provides an aggregated view of Bright data across data sets. So what the objective here is to, again, that automatic manner, high quality data gets seen across price data set for all users. So this is not a data set centric view. This is an across data set view. The pipeline starts with data coming from complete submissions. Those are ones that were standard file formats are available, MCR, MTML, and MCTAB. I'm not going to take to talk into account in detail about all the details, but there is a approach to group peptides and proteins. You have to take into account that we have millions and millions of evidences. I just said recently, he thinks that we are now close to 1 billion PSMs, so there's a lot of infrastructure needs to be done in order to handle that properly. And then there is a QC step and a protein inference step that is done using the P algorithm. And then what we have at the end is an aggregation of data per peptide and protein pair across data sets. Again, this is an automatic pipeline. This is how the web interface looks at the moment. This is work in progress, so I think it will be very improved in the next month or so, but it's already, you can see a lot of nice things there. And for each representative peptide, there is also a representative spectrum, which is the best scored one. Okay, so summary, just to finalize, I hope that you could see the increasing amount of data in the public domain. We have been doing that over the years with Bright and Protein Exchange. I think what we really need to have Proteomics data better represented in bioinformatics resources that are accessed by non-experts in proteomics. We are doing efforts based on data analysis in Spatial Atlas, UniProt for post-translational modifications, and Ensemble. Without data analysis, but with just taking the high quality data in a way across data sets, you can access that using our new resource, Bright Peptidome. I just need to finish thanking everyone in the team. So this is a fantastic thing. I want to highlight again the work done by Jacet, Matthias, and Anna. These are the kind of main highlighted studies that I have presented today. And of course, thanks a lot to all our funders and all our collaborators. So yeah, thank you very much.
Thank you very much, Jaun, for another excellent presentation. So we have a large number of questions here that I'm trying to get into some order at the same time as speaking. So I'm going to take some from the audience first, and then perhaps come in myself a little bit. So first question for Ollie from Harry Whitwell is, how do you identify bad quantitative peptides or features early so they aren't included in the protein quant value?
Yeah, that's a good question. And I guess, everyone has their own way of doing this, and they've decided, what's robust for their pipeline. My philosophical perspective on this is that kind of bad is a sliding scale from good to worse, and ultimately just drawing a threshold somewhere is deciding everything on one side is bad, and on the other side everything is good. And that feels really limiting, because I think there's a gray area in which we should be bringing some of these peptides through, but asking the question, well, what is the probability that peptide is good? And then use these probabilities as weights in whatever summarization that you use. So if you take all your scores that tell you how good your peptide is, and and say summarize them all up at a protein level using a model, you could say, well, this peptide looked like it was there was some precursor mixing, or we weren't sure about the identification, or the posterior error probability was high. Then you might weight this down in your analysis because you don't think it should contribute that much to the protein analysis. So hopefully you can follow up if any of that was unclear. That's how I would philosophically like to do it. Getting there mechanically is proving quite difficult. You know, if you're on the side of identification and you know, if you're doing a non-Bayesian analysis, how might you go about identifying which proteins are bad and which are good? My perspective is some kind of stability or robustness. So set different thresholds and see how your results change empirically. And then you can kind of say, well, why did my results change when I included a particular set of peptides or set a particular threshold? And biological results, which are kind of very sensitive to our choice of threshold, are the ones we've got to be kind of concerned about. And the evidence may go one way or another. But hopefully probability would allow us to kind of quantify these things a bit more exactly.
Yeah, no, I agree. I think for quite a while there's been this notion of proteotypic peptides that more often than not can be unique to an individual protein, and a few groups these days are moving over and thinking a bit about what they call quantotypic or peptides that can potentially themselves quantify proteins. Now, can they do universally? Possibly not. There's a lot to be got from data-driven methods of, say, as Ollie says, of estimating it from the data of an experiment. You possibly can borrow some strength from a larger contingent of, or even like Pride Atlas or something like that. But yeah, it's a challenge in an open research problem itself. So yeah, great question. Segueing to something slightly similar to Jaun, but probably more at the protein level. So I had a question on how do you quantify protein expression in your gene expression atlas? Now, I know you use established pipelines. The question really goes into how do you reflect any post-translational modifications or splice variants? And I guess my add to that was, how challenging would it be to do so if you don't?
So we are not doing, as I mentioned, one of the criteria that we are following at the moment is not to use PTM-enriched datasets, just to avoid the complexity. And in case of isoforms, I mean, we are using as the search database the UniProt complete proteome, but at the end we are converting the protein identifiers into gene identifiers, because basically the gene expression atlas is gene-centric at the moment. So in order to have protein expression and gene expression in the same kind of reference systems, we have to convert it to that. So just to highlight then that the complexity of the data that is seen in expression atlas is reduced in that context. So I don't know if that answers your question. For proteoforms, I think we are very far. I think that for that we need to do. We probably need to do a lot of things in the future related to top down, probably, or middle down. Or I think we are. I wouldn't. At least my opinion would be that we shouldn't get into quantification of PTMs, at least in this context of activities that we have been talking about. There are also not so many public data sets that are kind of suited for that purpose. So to compare between different conditions properly about the amount of modifications and things like that. So
I was just wondering, Jaun, how uncertainty might be specified in your bioinformatics
for
resource.
Sure. I'm sure we can discuss with Ollie a lot about this.
Yeah.
A lot of Yeah, there are many points of uncertainty, of course. It's,
is it based on quality control at the moment? You just put, so for instance, when you're integrating the gene and protein expression, the protein expression you put in, is that based on thresholds and what you consider a good quality?
Yeah, of course. I mean, but we are not using anything that is kind of very different to what other people are using. So it's something relatively, relatively basic. So I don't know, at least 2 unique peptides per protein. I mean, these kind of thresholds that are kind of used by many people. We are not reinventing the wheel there.
I wonder if I could bring that question over to Ollie, and particularly in terms of what challenges you think that to make Bayesian analyses or uncertainty accessible to researchers. How do you think you might represent that uncertainty if you're putting it into your results into a resource?
I mean, this is a very challenging question.
Sorry.
I think I'll preface this with I don't think I have any good ideas at this stage, but ultimately, we would love to be actually visualizing ways of, when that summary was done from peptide to protein, kind of what were the range of values involved, what was the distribution of that quantitation, and that's something that you could, visualize in your analysis, or any downstream results, there was, and what uncertainties were actually kind of incorporated. Some examples from our end and stuff in the Lilly lab, we do have Shiny apps where you can click on a protein from our spatial proteomics data set, and it will bring up a posterior distribution over the particular localizations. And I think this is like one step towards a good visualization of the results. But I think a lot of cases you're going to have bespoke visualization and bespoke communication of uncertainty, because what these probabilities means are very embedded in the model that you use. And so the data don't mean anything without a model, and that's all got to be communicated. Ultimately, it's hard for me to see the community being ready for all of this. I think there's kind of a slow period that we need to build up kind of good examples in the literature of where this is working and where this is not working, what's difficult to communicate, what's not, and then ultimately kind of converge on something that's useful as people get more comfortable with the idea of reporting an uncertainty or actually using it in some of their own analysis.
And just a follow-up, just specific to what you're saying in your talk. I think at the end of your talk, I think for the HD exchange, you were talking about expressing as like probabilities, I guess posterior probabilities, with a lot of experimentalists being very comfortable with p-values, and we're going into the age-old thing of how I mean, it might just be the same as the last question. How do we as Bayesian researchers, how do we get across of how the results should be expressed for the field, and would you express significance, or even should you express significance?
Yeah, that's a good question. I think the proteomics community is crying out for statistical standards initiatives, not just experimental standards and database standards. I think it's very unclear what people should report, a bunch of p-values, adjusted p-values from what method with what settings. I think even a frequentist framework, this is missing. I think in a Bayesian analysis, there's no need to report any kind of significance or thresholding. You just report your probabilities, and then you interpret them. And that's it really. Ultimately, people do want to kind of pick a threshold because they say, well, we're going to take these proteins from downstream results into, say, Go enrichment. And ultimately, then that leads us to a set of new questions of how do we incorporate these probabilities into downstream kind of testing results and enrichment and methodology, rather than just here's the things that I was interested in. So I think it opens up a load of new questions about methodology. I would love to just, I think a p-value is useful if you use it correctly, but I think it's very easy to misuse, and that's the problem. And the Bayesian framework actually forces you to do the job well. And that's something that's kind of not so apparent when using p-values. But in all frameworks, I think people should tell us what they're doing as much as possible, so that we can, there is better communication on all sides.
I 100% agree with you regarding a statistical standards and I guess minimum reporting, that kind of thing. That would be a fantastic idea. I've got, while we're still on the subject of quantification, I have a couple of questions here from Karin Banuin. Sorry if I didn't pronounce your surname correctly. Jaun, and I had a bit this question too. How are you normalizing across data sets? I mean, presumably within the various data sets you're using, there might be biases and batch effects, etc. Could you explain a little bit more about the normalization you're doing?
So I mean, there are two ways to do this. The usual way where that people have used over the years is to analyze all data sets together. No? So we wanted to do a large meta study of different data sets, then one, put together like, I don't know, 20 I don't know, even 100 data sets to try to improve the normalization across. So we have done that in the past, but the problem is that this approach, it is better in terms of the consistency of the results, but then it's not sustainable. Because, for instance, if you want to add tomorrow an extra data set that you found that is interesting, then you cannot, do the same reanalysis again with 100 plus 1 data set new that was there. And this is really not sustainable in medium long term. At least it can be for a research project, but it's not sustainable in a production kind of environment. So what we have tried in this last year is to do the analysis independently, so data set per data set. This, of course, creates effects across data sets. This is kind of unavoidable. It's really unavoidable. So what we try to do in order to make the results more comparable across is to simply go using the ranges of abundances from in each data set and separate it in different bins. So we have used kind of different amount of bins in order to optimize this. At the moment, we are using 5. So that means that intensity values between across different datasets are separated in different bins, and that helps to make data more comparable. So I didn't have time to explain this in detail, but of course there are many limitations on top of that. It's far from perfect, but at least it helps to reduce a little bit the problem of the batch effects. There are other methods to do that, and we have also tried that in the past, like performing PCAs and these kind of things. But I would like to highlight that if we want to do what I have explained, in a kind of production environment that data sets are reanalyzed kind of semi-automatically, there is no way to do this if you don't do this in kind of data set per data set. And this is, for instance, the approach that was taken by our colleagues in the analysis of transcriptomic data sets a long time ago. So I don't know if I answer your question.
I don't know. We'll see, but yeah, no, I think obviously the heterogeneity of the data that you have in PRIDE is very challenging, but you do have the advantage of having a lot of it and a lot of studies, so that you would hope that over many studies that a lot of this variability and bias may average out.
Yeah, that's the idea, of course.
Another question from Karin, this time for Ollie. Have you used Bayesian stats to identify significantly different abundance proteins in proteomics datasets that have no enrichment steps, just, I guess, standard global proteomic profiling? If so, how well does it work for you?
Yeah, it's probably more Andy's territory, and there's probably kind of a dozen or so methods kind of trying to do this, and I've played with some and then try to write some of my own just to play around with, see what happens, and in general they work well. The easiest one I found to use has been Trickler, which has worked very well. I'd say with a lot of things, when you start moving to a Bayesian analysis, it opens up the fragility of the method, and you realize that not one model fits every data set. And this is where the challenge comes in, is that every data set will need its own kind of modeling and parameter optimization and fiddling around. So I think that's definitely something that us Bayesians need to think about carefully, is how do we help people with the kind of model building process for their data set, because I don't think one model fits all, even within the kind of proteomic profiling field where everything feels somewhat similar. But maybe Andy has some comments here.
Yeah, in fact, I've just been given permission on the chat to talk about the work we've been doing. So we have a tool called CMAS that we've been working on for quite a while, which is Bayesian tool set for standard discovery experiments, and we've been trying hard there to make it accessible in terms of giving you the same kind of feeling and results as a standard frequentist approach. I think that the Bayesian analysis is really useful, because you can add the models can be more complex and still you can solve them. So that's what works out. The other thing that's actually quite good is that with a tiny bit of hand waving, when you come to false discovery corrected p-values like q values, the Bayesian versions of those are basically the same thing, and they can be interpreted in the same way. So while posterior probabilities and p-values are a bit different, both can be converted into false discovery rated q values basically, and interpreted in the same way. So hopefully that will mean that our work, Ollie's work, my work might get a bit more traction in future. Right. Of course, now while I was explaining that, I need to keep a tab on the questions coming in. So let's change tack a little bit. This is a question for Jaun from a Joe Kup. I don't know if I pronounced that surname again. Sorry. One of the most, so it's about the challenge in pride of finding metadata surrounding the files that are submitted. I know that actually I've heard you talk about this before, Jaun, but sometimes the metadata is missing altogether, and I don't think that a lot of it's required. How do you think this could be best addressed, short of relying on the submitter to have the intention that their data can actually be researched or mined meaningfully?
So, I mean, this is a problem that we have known from the beginning. So when one has to look at this with some perspective, when we started, Proteomics Change 10 years ago, the objective was to put a lot of data in the public domain, because there was really very little at that point. So we received feedback from many people saying that, the metadata was kind of not that important at that point. The important was to have a lot of data in the public domain. So this is the direction that we follow, and we realized that the lack of metadata is really a problem. I mean, we are the first ones that we are experiencing this, as you can see. So this problem is also not that simple to solve. Something that I haven't had the time to talk today, because the focus on the presentation was really on the reuse of data. It was an initiative that has been led by Jacet from my team, but also EUBIC researchers, and is the development of a data format that is kind of a standard to provide metadata for proteomics experiments. Actually, the paper came out earlier this week in Nature Communications. You can see this. I could put the link later in the chat, but it's a format that has been developing in order to enable that. So now we have a format. Now we need the tools to produce that format, and also we need the willingness of the submitters to provide that information. So so these are kind of two different problems. The tooling problem is one of them, and I think that for that, only a single team like Pride cannot really solve the problem. We need to rely on people like develop tools in the community to produce this format as well. And the other part, that is the the submitters, they need to you need to kind of incentivize that they provide the needed metadata without making the submission process too troublesome. Because if that happens, basically what will happen is that that people will not perform the submission, or they will instead go to other resource. So this is kind of a problem that we are aware, and this is kind of the direction that we want to go. So we have now a format is made stuff for proteomics. It has been we have reused what our colleagues in EBI have done for transcriptomics before, and as I said, now we have to have the tools that produce this file. Actually, we already support in Pride. I haven't mentioned that if people are providing this file, we can already receive it, and it can be visualized in the web interface, and also it can be made available. But not many people have done it so far because it's kind of optional. But yeah, these are the only way to improve this in the near future, is to incentivizing people to provide this format.
As a follow-up, Ollie had a question himself, so maybe he'd like to expand on it, but he was interested to know how much public data is currently being reused to obtain new or overlooked insights.
So of course I cannot read all the papers to know what is new, but what I can tell you, for instance, is you saw the slide with the volumes of data that gets downloaded. So of course that doesn't mean that it's reused. And what I can also show, I can put that slide, but there is an increasing number of papers every year that include kind of reanalysis or reuse of data sets from Pride. So this can be checked manually. I do it regularly, and this number of papers has increased over the last few years. So in 2021 so far, there are like 100 papers. But this is, of course, an underestimation, because it's just the ones that we can really detect directly. And sometimes it's not easy, because even if you want to do this automatically, it is not possible, because not all papers are like open access. And then, because if you would like to do this automatically, you need to have the possibility to do text mining, looking for identifiers and these kind of things. So this is an approximation. The other approximation is, there are again resources that are kind of popping up, like the ones that I have mentioned, but there are others, and that that they are highlighting proteomics data more and more. So it is really difficult to quantify exactly, but it's really increasing. That is for sure. So yeah, I don't know if I do like to follow up on that.
Thank Ollie, you okay? Or
yeah, no. I was curious if you had any favorite examples of people using the resource. And
so, for instance, I mean, there are yes, there are some studies that are using now public proteomics data and machine learning approaches in order to have new findings. I can, if you want, Oli, I can put a few examples in the chat later as well. I can put it there. It's probably easier.
Yeah, sounds brilliant. Thank you.
Right, we're coming up near the end. I feel though that I should ask you one question each. So if a short response, if you can, to these. Firstly, from Alessandro in the audience. Juan, how do you handle protein inference across all your study data sets?
So so again, this is the same thing that before. If you do the reanalysis of all data sets together, you handle it together. If not, there is not a way that do it data set per data set. But this is less of a problem if you have some kind of assumptions. And as I mentioned before, also at the end, the proteins are often converted to genes anyway, at least for the purposes of showing protein quantification in spatial analysis. For other things like PTMs and yeah, we are handling using the kind of the way protein inference is handled in those tools, in the tools that we are using basically. So yeah.
Great, thanks. And Ollie, this is actually a question from me. I was interested when you're talking about your semi-parametric model, and you talked about deviation from the sigmoid. I guess how do you define the deviation? And together with that, and maybe I was misunderstanding the spatial proteomics and subsegment underneath, but is there value, and maybe you do, cluster your curves together to see if there's types of curves that come up regularly?
Yeah. So I think I'll answer those in reverse order. So the model that underlines the spatial proteomics is basically a kind of semi-supervised clustering model. So ultimately, there is clustering of proteins together that belong to the same organelle across kind of each of the data sets. So that's built into the method. And then in the semi-parametric model, you can ask explicitly, is my sigmoid model or my semi-parametric model better at explaining the data? You can say, oh, what is the probability of the data given model, semi-parametric model? What is the probability of the data given sigmoid model? And just either compute the ratio or some kind of posterior probability for that. Or you can look at some kind of more predictive-based methods. So you can say, oh, if I remove data and predict it using these models, kind of which ones do better. So you can ask both of these questions kind of directly from the distributions. I think we've lost Andy. But
yeah, we can hear you.
You're muted.
Okay, right, here we go. I'm back. Thanks very much, Oliver. That answered that very well. So that's great. So it just ends for me to thank both of our speakers again. That was a great discussion section, and now I'll give back to the host, Joe KP.
Great, thanks, Andy. Yeah, so as mentioned earlier, we will have a form for those who require a certificate of attendance, and that will be accessible for a few minutes after we wrap up. I'd also like to say thanks very much to both of our speakers and the guest chair for really lively discussion, and for everyone for coming along, and to all those committee members who are working away in the background to make this webinar possible. So we are planning to be back in November with a hybrid virtual and hopefully in-person event in London. So keep your eye on our website and social media for details once we've got a programme finalised. In the meantime, we wish everyone a great weekend. Thanks very much, everyone.
Thank you.
Thank you very much.